This course is on scientific data visualization with biomedical applications, using the tools of bioinformatics and systems biology. You are now watching the second section of the lecture. Our first topic is to describe the picture as a visual message. We will now talk about a scientific picture as a visual message. So first, what is a message? We use the classical definition of Claude Shannon, often called the father of information theory. As we see on this slide, a message has an emitter, coding and decoding processes, and finally a receiver. In Shannon's case, the message was a series of 0 and 1 bits sent and received by telegraphs. In our case, the message will be the scientific picture, a figure of our thesis, etc., which is sent by one person to the other. Let's now apply the message principle to a scientific figure or slide. The emitter is the person who draws the picture. Coding means the program you run to create your picture. The filters can be different in various situations. For instance, the resolution of your picture is an important one. There is a filter on the receiving side too. This is the resolution of the reader's computer, or the resolution of the projector in a conference room. The receiver is the audience of your presentation, the readers of your manuscript or dissertation, the reviewers. It is clear that we need slightly different pictures for different situations and audiences. And finally, decoding happens in the mind of the audience. Make sure it does. For instance, an audience should be given enough time to read and understand the message and send it to the long-term memory. This is now a written summary of what we said about the picture as a message. In general, a live audience has no time to read such a slide, but if you make a handout, such lists are okay. In other words, even if we want to send the same message, we need different slides for different occasions. Let's see what these occasions are. First of all, who is the audience? We can speak to students, conference participants, or show our figure in print, on web pages. All these require somewhat different figure styles. Namely, we do want a message to go through, so the target audience must have sufficient time to read it. If we speak to a live audience, we cannot put too much text on a slide. Rather, we need simple slides with clear emphasis. In contrast, readers of a journal have more time, so a figure can be more complicated. However, readers and opponents can easily get irritated if the figure is too complex or if they need to look up information in other places. It is a useful rule that a picture must be understandable in itself, that is, it should be a standalone message. A picture as a visual message is always a part of something. Generally speaking, it is a part of scientific communication. Some of the forms are listed here, like scientific articles, posters, presentations, etc. Going into detail here would need a separate course on these subjects, such as article writing, etc. Here we only point out that most of these presentation forms have multiple audiences. For instance, a scientific article must be understandable for the readers, but it also need to be accepted by the reviewers who may have quite different preferences. Also, a diploma thesis can be sent to reviewers with opposing views. So we need to design figures that satisfy all these sometimes conflicting requirements. Second, we often have multiple communication channels. For instance, an article has text and picture. A slide presentation like this one has picture and audio channels, and the picture channel has both text and figure. So our visual system has to do its best. The strength of the visual perception system is that the failures of vision can be corrected by the cognitive functions of the brain, and vice versa. We can imagine this system as a complex filter. Information goes through only if vision creates a picture that can be correctly recognized by the brain. So we can accept even a blurred picture if it retrieves the correct item from our long-term memory. Troubles come on when we have no background knowledge matching the incoming picture. And this happens quite often in scientific lectures. For instance, we do not see the screen very well or we do not know the actual field very deeply. When this happens, we say that the speaker has lost the audience. By the way, are you still here? First of all, we should realize that a picture should have an understandable focus, or as we say it sometimes, a story to tell. In many cases, the story is a conspicuous pattern that the reader can easily recognize. In simple plots, such a pattern is a peak. In scatter plots or in networks, the patterns are clusters that one can easily recognize. Adding emphasis to figures is an old trade. Arrows, labels, colors are the most useful tools. Take a moment to look at these figures. Diagrams, scatter plots are examples of simple data visualization. If we show distributions, we add interpretations to data, and so we communicate information, not only raw data. Finally, we can make a taxative statement, such as two populations are different. Combining all these as shown here illustrates the story of a project, like a graphical abstract. Visualizing conclusions only is more like infographics. These subjects will be talked about later in the course. Graphical abstracts are relatively recent figure types. Essentially, they are figures summarizing a project without necessarily showing the data. Many scientific journals request graphical abstracts that are published together with the article. In informatics, the well-known flowchart diagrams are a kind of a graphical abstract of a computer program. In the other sciences, summary figures have been prepared from the earliest times, but the graphical abstracts today are more organized in the sense that they have strict rules prescribed by scientific journals. The preparation of such figures is facilitated by libraries of dedicated icons, such as provided by the BioRender program presented later in this course. Finally, let's remember, many of the important discoveries in science were presented in a visual form. Darwin's tree figures are symbols of the evolution. Mendeleev's periodic table elegantly summarizes the ground rules of naturally occurring chemical elements. Finally, the double helix of the DNA opened a new era in molecular biology. It shows a structure that could be drawn only in this simplified form, so it is a real masterpiece of simplification. We can of course argue that these pictures became emblematic because of the scientific advances they created later. This is of course true to some extent. But at the same time they clearly show that visual messages are far more important than many of us think. Now we turn to the fourth subject of our lecture, mapping data to templates. First, let's see the four major groups again. General statistical data, molecular structure, networks and genomes. The first group of programs includes general-purpose software that we can use to produce diagrams used in various fields, especially statistics. Why do we discuss them here? The answer is simple. Many of the analysis types in bioinformatics or medical informatics are nothing but simple statistics. This is because the complex objects of bioinformatics, like genomes, networks, macromolecules, are sometimes described by simple numbers. For instance, the number of mutations, the nucleotide composition of genomes. And these numbers have to be analyzed by reliable statistics, which is a mandatory part of bioinformatics papers. This is why we devote a part of this course to visualizing statistical data in bioinformatics or medical contexts. Genomes represent a central data type in current bioinformatics. This is why we included them as a separate part of our course. Genomes are essentially linear structures that need to be simplified so that we could discuss them. And then we often add additional info so that we could emphasize a certain point. We will show a genome mapping example later. Three-dimensional structures of proteins and DNA are essential for explaining biological findings. It is enough to think about coronavirus infections where three-dimensional explanations of the infection mechanisms became a daily part of the news in popular journals. This field is somewhat different from the rest of bioinformatics since the three-dimensional templates themselves come from experimental results, such as NMR or X-ray crystallography. An entire section of this course will be dedicated to three-dimensional visualization. Finally, networks represent perhaps the latest arrivals to the visual zoo. From the logical point of view, everything in biological informatics, from simple molecules to genomes, can be described as a network. But more importantly, there are hot topics, for which networks are the only plausible descriptions. It is enough to think about metagenomes, microbiomes, drug-target interactions, protein interactions, etc., so that we realize that network visualization is essential for most of the currently studied topics. Later in this course we will have 2 lectures on network applications. Now let's see the details of mapping, first in one dimension. A one-dimensional template is like the x-axis of a coordinate system. Mapping a single value on this axis gives a line graph, like a simple function. Here the diagram on the right has one important message: we have a conspicuous peak, a non-random feature. Examples of 1D analysis include time series in engineering and medicine. For instance, we can think about electrocardiograms, etc., which is outside the scope of the present course. And of course biological sequences in bioinformatics, such as proteins, genes and genomes. Here we see that we can have an arbitrary number of variables in our data, and we have to make a choice how we plot them. Let's plot 2 variables as a function of x. This gives us 2 line graphs, and in this particular case we have a simple message: v1 has a peak, v2 does not. We can also plot the 2 variables against each other. The result is what we call a scatter plot. The plot on the right has a simple message: our data cluster into 2 groups. This is something that we do not necessarily see from a line graph. This example shows you how we turn a genome sequence into a picture using a one-dimensional representation. Naturally, we do this only in a very simplified form since the operation is a full bioinformatics project, called genome annotation, so we will not go into details. Also, understanding this slide requires some familiarity with bioinformatics. In any case, a full genome consists of several thousands to several million nucleotides, so it does not make a good picture. In the first step, we replace it with a straight line that has no details. This is shown above, and we will now follow the arrows pointing downwards. Then, we use a gene predictor program to identify the genes that we add as boxes to the genome sequence. Again, we cannot show it for a whole genome, so we show here only a small part of the genome. In the next step, we add the names, functions of the genes using database search. This is indicated by callouts. Finally, if we have quantitative data such as transcription intensity, protein binding intensity, etc., we can add them as diagrams, as shown in the below picture. This is quite a complex figure, so if we want people to understand it, we can only show small parts of the genome. We have dedicated programs for this that called genome browsers. In this picture, we use an annotated genome as a template to visualize the hypothetical effect of disease mutations. Let's say the genome is the fully sequenced human genome on which tons of data accumulated over the years. This will be our reference genome.
Let's now suppose that in a patient we discovered 3 mutations, A, B and C, that we will map on the human genome. The resulting picture can be translated into the following message. Disease-associated mutations A, B and C map onto gene X, both to the coding region and to the putative promoter that harbors a transcription binding site.
This may not sound too much perhaps, but this is a publishable message which is illustrated by a simple figure. Also, this is an example of how knowledge is accumulated in a database. The reference genome is a database entry to which we add disease-related mutations. Now we turn to mapping in 2 dimensions. A 2-dimensional template is a part of the XY plane. Plotting a value onto this plane can be imagined in 2 ways. Either we plot a variable as a 3rd dimension, in this case we will get a 3-dimensional object, a surface. Which may be nice, but we may have to play around and rotate it in iterations so as to see all the details. The other solution is to encode the variable as a scale of colors or a shades, and plot it to the plane. The result is what we call a heat map. This is perhaps less attractive than a 3-dimensional surface, but it can be understood without rotation. A hidden take-home message of these last slides is that an interesting picture is one where we see a non-random pattern, like a peak in 1 dimension or clustering in 2 dimensions. Now this is a classical example of how to find clusters by using simple templates. These are hand-drawn, pre-made templates that correspond to the top view and side view of an idealized alpha helix, respectively. Usually we choose sections less than 20 amino acid long. We write our protein sequence into the template in the required serial order. Then we color the amino acids. In this case we color the charged with one color, not charged ones with another color. If we are lucky, we see clusters, like in this picture. Both figures show that charged amino acids are on one side of the helix, non-charged ones are on the other. What does this mean? Peptides that have this property will bind to cell surfaces with the charged face pointing outside and will eventually disrupt the membrane and kill the cells. Antimicrobial peptides have this property. So if we see such a peptide sequence, say in an insect proteome, we can suggest that the peptide is antibacterial. It is important to note that the message was based on an asymmetrical pattern, same as we saw with the previous examples of 1D plots and scatter plots. More exactly, on both figures we see the coincidence of 2 asymmetries, one in sidedness, and one in charge. Networks are peculiar in the sense that a mathematical description does not tell us how exactly to draw them. For instance, we can introduce long edges that allow nodes to be pulled out far from the rest, still the mathematical description will remain the same. In other words, we have to apply extra knowledge and usually some kind of optimization so as to draw a compact network. For instance, we can look for the short connections with a minimal number of crossings or other non-biological criteria. We will have a separate lecture on this topic. On this slide we see a hypothetical protein-protein interaction network which was first drawn with the default settings of a network visualization software. This gives us indications of 3 clusters. In the next step we colored the nodes according to their biological functions looked up from a database, and found that the 3 clusters are indeed different also in terms of function. Note that here again we see the coincidence of 2 clustering patterns, one in connectedness and one in biological function. Macromolecules are three-dimensional structures, but we can plot additional data on them either by assigning colors to various elements or by using the heat map approach, assigning colors and shades. On this slide we see the structure of the acetylcholinesterase enzyme in three different views. On the left panel we show a conventional structural view. Here the template is the main chain or backbone, which is indicated with a red arrow. On this barebone we add three-dimensional symbols corresponding to idealized alpha helices. These idealized ribbons then give a realistic-looking view to the structure, but this view is purely symbolic and not at all realistic in the physics sense. Panels B and C show a surface view of the proteins, which is a computed surface. In the middle, the surface is colored in a heat map fashion according to hydrophobicity. Highly hydrophobic amino acids are colored red. On the right panel the surface is colored according to the charges, red is positive and green is negative. We can notice that the two right panels are complementary to each other. This comes from the fact that hydrophobic amino acids are not charged on vice versa. The previous examples give us a few take-home messages summarized in this slide. First, data are collected with complex techniques, e.g., sequencing, X-ray, NMR, and even though we deal with visualization programs in this course, it is a good idea to have some basic understanding on data collection methods, for instance at the Wikipedia level. Visualization programs may directly produce a picture file. However, in most cases we need further intervention, adding three-dimensional perspective, rotation. And this iterative refinement is the heart of data visualization. This optimization is carried out by a human operator like ourselves, and this is often a tedious iterative process. So one of the most important practical ideas is to make pictures in such a way that we could modify and redo the pictures with a click.